morning, everyone. My name is Pei Wang. I'm a professor of genetics and genomic sciences from Icahn School of Medicine at Mount Sinai in New York, United States. First, I want to thank the meeting organizer for the nice invitation. It's really a great pleasure to have this opportunity to participate in the Zero Childhood Cancer National Symposium and being able to share some of our latest research work with the audience here. So today I will talk about proteomics, genomics analysis of pediatrics, adolescent and young adult brain tumors. So in this talk, I will first give some background about a nationwide effort. It happened in the United States, led by NCI and CPTEC consortium to using integrative proteomics genomics analysis tools to study various types of cancer. And then I will talk about two studies, which both comes from CPTEC and collaboration with other consortiums in the United States. The first study is a published work focusing on using integrative proteomics genomics characterization to study 7 major pediatrics brain tumors. And the second study is an ongoing study, which is further extend the previous study of high-grade glioma in pediatrics to cover adolescent and young adult age segments. Okay, so first, I think by now it has been well agreed that actually genomic analysis do not reliably predict alterations in protein expression or function. In the past decades or 2 decades, the technology of proteomics profiling has really been improved. So we are able to obtain the protein abundance measurement and PTM abundance measurement in a large scale genome-wide manner. And from those studies, we noticed or learned that actually the correlation between genomics data and proteomics data is much lower than we expected. For example, if we think about mRNA expression level versus protein abundance level, the median correlation of the same gene is actually only 0.45. This is some number. This number is consistent across many studies of different tumor type, different tissue, or different disease. And if we further think about PTMs, like phosphoproteins and glycoproteins, then the variation of the activities in PTMs, the percentage of those variation that can be explained or predicted based on genomics data is really limited. So this raises a great challenging for precision oncology, which has mainly, focused on genomic analysis by now, because the modern therapies are usually being designed to target proteins. Therefore, a better understanding or characterization of the protein activities actually would be very, very crucial. Okay, so to address this challenge, in the United States, the National Cancer Institute launched this CPTEC, Clinical Proteomics Tumor Analysis Consortium, to really push forward and enhance the proteomics profiling and application of those on the cancer studies. So this effort actually started quite early, back to 2004. And in the first 7-8 years, the effort is really focused on technology development and technology validation. Starting from 2011 to 2016, we are able to apply the proteomics high throughput proteomics profiling to a subset of the TCGA tumors and demonstrated that, yes, the proteomics data as valuable information that is not observable in the genomics data. Therefore, NCI really invested a large amount of effort and resources and has been carry out large-scale proteomics genomic study for a large collection of tumor types. So this is so in the phase 3, CPTAC phase 3, in the past 5 years, we have studied almost 10 tumor types, and in the coming 5 years, which will be CPTAC 4, we will study another 6 to 7 tumor types. And in addition, besides discovery studies, also the consortium has trying to push forward of this application of proteomics analysis in clinical trial settings. Okay. So as I mentioned, it has been demonstrated that by adding proteomics data to the genomics data, we gain a lot of insights. So during the CPTAC 3, the consortium or the research team working together and really contributed a large amount of valuable proteomics genomics data of many, many different tumor types. And we, based on those data, we generated a lot of hypotheses that hopefully can lead to new therapeutic treatment or drug targets for those patients, for cancer patients. Okay, specifically, there are 2 studies from CPTAC 3 that is focusing on brain tumor. One is the adult GBM, and the other is a pediatric brain tumor study that covers 7 different histology types. And actually, this work is a great collaboration between CPTAC and another 2 big consortiums about kids cancer. One is the Children Brain Tumor Network, and the other is the Kids First. So this is really a great collaboration across many research teams working together to conquer brain tumor. And in CPTAC 4, we will further continue those great collaboration to do a few more large-scale studies, including proteomics genomic study of about maybe 400 more tumor samples of pediatric brain tumor, of others, 4 to 5 different histology types, which is just launched. And then we also have our ongoing project, which is extending the previous HGG proportion in the pediatrics brain tumor study, but trying to cover not only pediatrics, but also adolescent and young adults, such that we can link from the pediatrics end to the adult end. And we also have another adult brain tumor project ongoing. Okay, so today I will focus on these 2 projects. The first pediatrics brain tumor project, as well as the current ongoing pediatrics and AYA HGG studies. Okay, so pediatrics brain tumor, maybe many of you guys are already very familiar with this, but just in case there are audience not familiar with pediatrics brain tumor, this is actually the leading cause of disease-related death in children. The current classification of brain tumors are based primarily on microscopy morphology and IHC type of technology. So we really need maybe better precision medicine approaches that can help us to understand the development of the brain, the cancer, and have better treatment for those kids. I think in the past, we really, because of the brain is in a different developmental range, so the pediatrics brain tumor actually represents very different disease mechanism to adult brain tumor. Okay. So one special characteristics of the pediatrics brain tumor is that it actually has very much lower mutation burden than adult tumor. So as shown in this figure, the left part are the mutation kind of burden in the pediatrics brain tumor, in the pediatrics brain tumor and other tumors, while the right hand is the adult tumor. And then you can see apparently there is a much higher mutation rate in adult tumor than in adult tumors than the pediatrics tumors. So this really motivated us to leverage the proteomics data to have better understanding of the therapeutic strategies of those pediatrics diseases. Okay, so this is how the motivation we carried out this pediatrics proteomics genomics characterization study. Okay, So before I jump on the science, I want to really acknowledge all the research collaborators in this project. The collaboration is really key, that the project involves like 20-ish different institutes. And also I want to thank patient and the patient family for contributing the tissues at the first place, such that we can make this project feasible. And also I want to thank like Brian, Adam, Philip, who are the co-corresponding authors, PIs of this project, and Dr. Amanda Pavlovich and Dr. Steve Kiki are leading the proteomics labs who generated the proteomics data. Okay, so here is a highlight or summary of this proteomics study, proteomics genomic study. So first, this is the first large scale proteomics genomic study of pediatric brain tumor. We actually profiled more than 200 pediatric brain tumors spanning 7 different histology types. And then, based on starting those data, we identified new treatment strategies for some of the cranial pediatrics patients. And we also identified new prognostic markers for hybrid glioma in the pediatric brain tumor patients. And by comparing initial versus recurrent tumors, we found that the recurrent tumor are really different and probably should be treated as different tumor in the context of the targeted treatment decisions. And we also developed a data portal that can enable users to query, visualize, and download the data coming from this study. Due to the time limitation, today I will focus on the story of this second highlight, which is a new treatment strategy for cranial tumors in the pediatrics patients. Okay. So first, let me show you an overview of the study cohort. As I mentioned, we studied more than 200 pediatric brain tumor samples, and among them, actually, we have 18 paired samples that's including our initial tumor versus a progressed tumor from the same patient. And for every tumor, we performed the genomic profiling, including the whole genome sequencing, RNA-seq, as well as the proteomics profiling, which included global proteomics and phosphoproteomics. In the global proteomics data, we can, after QC and every pre-processing, we are able to reliably measure the abundance of almost 9,000 proteins and almost 18,000 phosphosites in each sample. And the left side illustrated the decomposition of the sample sizes in each of the 7 histology types. For some of the histology types, it is pretty rare, so the sample size is very small, including the craniopharyngiomas. Okay, so on this slide, I'm showing a hierarchical clustering, a consensus clustering result using the proteomics data of the more than 200 tumors. And we are able to cluster samples into 8 different protein clusters as illustrated here. So as you can see, different clusters really have kind of very different protein activities across those 9,000 proteins. And among those clusters, there are very different interesting characteristics. Some clusters are represented by one histology type, like we have a very enriched of ependymoma cluster, and we have this column is actually illustrated the diagnosis subtype. So we can see the first one, yeah, is a very enriched ependymoma, and the second one actually is a very enriched medullary. But there are many other clusters. It's a mixture of multiple different histology types. And there are some histology types that has been distributed across many different clusters. So that is kind of interesting. And also, by looking at those box plots, we can see the stemness and the proliferation. And the RNA protein correlation actually differs across different clusters. And also the overall survival of the samples in the 7 clusters are kind of significantly different. Especially there are 2 clusters has very aggressive disease that the survival outcome is very bad. Okay, so then this is an overview, but I want to draw your attention to 2 out of the 8 clusters, which is cluster 4 and cluster 8, that both cluster contains a subset of the cranial tumors, which is this orange color tumor. So this is quite interesting. Why this is interesting? Because in cluster 4, this group of cranial tumors actually is grouped together with some of the LGG tumors that has this V600E mutation. And while for the other cluster 8, the cranial tumors are also clustered together with a bunch of other LGG tumors, but there's mainly representing BRAF wild type LGG. So why this observation is, first of all, before that, I want to comment that this pattern is only recognized based on proteomics data, including global proteomics data, phosphoproteomics data, and a kinase activity matrix derived based on phospho data. So in all of them, you can see that when we cluster tumors, cranial sample are separated into 2 different clusters in or 3 kind of clustering result. But in the RNA, we actually will not be able to see a separation of the cranial tumors. That all the cranial tumor is grouped in just 1 cluster. Actually, the RNA data are more, the clustering of the RNA data are more consistent, I would say, with the histology type classification of the tumors. Okay, so then, so why this subtype of cranial tumor is so interesting? So first of all, the craniopharyngiomas currently has no effective chemotherapy, actually. And there's really in a pressing need of something more effective treatment for the patient. And the BRAF V600E mutation, although usually not detected in pediatrics craniopharyngiomas, are often observed in the adult cranial tumors. So this is very interesting. And we know that there are successful treatments like MEK inhibitors, targeting those BRAF V600E tumors has been successful in the low-grade glioma tumor patients. So therefore, this gives us this hypothesis that maybe this MEK inhibitor treatment could be beneficial for the subset of the craniopharyngioma patients. And so we try to validate this from this pattern from 2 different angles. The first, we look at the kinase activity score derived from the phospho data. And the second, we also generated some validation proteomics data set using MRM platforms and trying to see whether the MEK pathway proteins are indeed upregulated in those subset of the cranial tumors that is showing similar protein activity profiles as the LGG BRAF V600E tumors. Okay, so first, based on the kinase activity score, using the phosphoproteomics data and using the current knowledge of which kinases is regulating which phosphosite, we are able to derive the kinase activity score. And this matrix is illustrating the kinase activity score of 40 cranial tumors. And the left part representing the cranial tumors that has been classified to the cluster 4, which is the cluster including the other low-grade glioma BRAF V600 tumors. And on the right is the other subtype, which is the C8. And we can clearly see that the kinase activity score is really very elevated in the C4 tumors compared to the C8 tumors. And so the diagram on the right further illustrated the key players of the MAPK, the kinases in the MAPK pathway, as well as the phosphosite regulated by those kinases and how they were up or down regulated in C4 and C8 tumors. As we can see, it's really consistent. And also, we can also see this AKT1, which is a significant player of the mTOR pathways, are also upregulated in C4 compared to the C8. Okay, so the conclusion is that the MAPK1 and MAPK3 kinase activity scores were both upregulated in C4 compared to the C8, suggesting that MEK inhibitor treatment might be appropriate for the subset of tumors. And we also performed another validation experiment to using independent MRM assays to measure the abundance of some of the key members in the MAPK pathway. And again, we can see that so the abundance of those key markers have very distinct abundance distributions in the C4 and the C8 tumors. And the MRM measurement is very nicely, show very nice concordance with those from the original TMT experiment, that is the global proteomics data. So this is further validated kind of the signals we see in the discovery data sets. And another interesting thing for the cranial tumor that we observed in the study is the immune landscape analysis illustrated that the cranial tumors not only is enriched of the epithelial cells, but also has very elevated abundance of PD1 and CTLA4. So this is motivated that a hypothesis that maybe their cranial tumors could benefit from the immune checkpoint therapy. Okay, so it's really based on those observations from the study, together with the collaboration with the PNOC, the Pacific Pediatric Neuro-Oncology Consortium. A clinical trial has already been launched to test whether the MAPK inhibitor and immunotherapy checkpoint treatment could be helpful to some of the cranial tumor cancers. So this is really exciting, and we are still keep collaborating with the investigators from the clinical trials to using proteomics technology to facilitate some of the assessment of the treatment or build up further clinical assays that can help to predict which patient might be more suitable for certain treatment arms. Okay, so that is the first part of my talk. And in the second part of my talk, I will focus on the second topic, which is the high-grade glioma study for the age span from pediatrics to adolescent to a young adult. So that is our ongoing project. Okay, so high-grade glioma, actually this is one of the histology type in the previous pediatrics project I mentioned. For your information, glioma are the most common malignant brain tumor in pediatrics and AYA patients. And actually this is a terrible disease, most times incurable. And despite of the effort in the past few decades, the survival time hasn't been improved at all. Especially we are interested in age span 15 to 40 because the patient from this age span has been greatly understudied and underserved on one hand. On the other hand, it has been suggested actually the AYA, the adolescent and the young adult HGG patient actually could have unique biology. To further illustrate this, we have built up our in-house reference database of HGG and GBM. That is a collection of data from the previous HGG and GBM studies which provides the survival outcome information of patient as well as the mutation information of the patient. Okay. So based on this reference database, if we compare the survival outcome for the patients in the AYA age span, you can see it's significantly better than the adult as well as the pediatrics. And actually, if you look at the survival probability at 3 years, actually, they're probably almost doubled in the AYA group compared to the other age group. So this is very intriguing, right? So is it possible that this is really a different disease? Or is it possible this is the same disease with the other age group, but because of the brain development is in a special status, and the interplay between the brain development and the tumor help the patient to have a good survival outcome. And if that's the case, if we can understand what is going on, what are those interplays, then maybe we will have a better idea how to treat this disease across a larger age span. Okay, and at the same time, if we look at the mutation profiles, we can see there are 3 groups of genes has very different mutation frequency profiles along age. Especially, let's focus on this first panel. As we can see for TP53, IDH1, ATRX, H3F3A, those genes has much higher mutation frequency during the AYA age span compared to the other age span. So this is another kind of evidence suggesting most likely the AYA HGG could have a unique biology, right? So in order to understand this HGG and the GBM across the different age span, the leadership from NCI, Henry and Tara, together with the leadership from the CPTN, Adam, Brian, and others, successfully established a great collaboration and to study the pediatrics and AYA HGG, and we call this study HOPE, which certainly is a good name. Okay, so this slide summarizes the HOPE cohort. So in this study, we have 95 fresh frozen tumors collected from 87 patients. And among those patients, about 2/3 are grade 4 glioma, and about 1/3 is grade 3 HGG. And at the same time, about 40% are patients coming from the AYA group, while about 60% are patients coming from the pediatrics group. We employed high throughput global proteomics, phosphoproteomics, whole genome sequencing, RNA-seq, and methylation profiling to study those tumors. And this matrix illustrated the availability of different modality of the omics data for each of the sample. At the same time, we also planned or have conducted some of the validation experiment by utilizing either the targeted proteomics platform or to leverage the single-cell RNA-seq data or microRNA data, trying to validate some of the patterns or hypotheses we observed based on the discovery data. So that is the scope of the study. This is an uncoprint summary of the mutation landscape of this cohort. While we can clearly see there are some of the genes has pretty high frequency of mutation in this cohort, including especially TP53, H3F3A, and the ATRX. So this is very consistent with the mutation profile we see in the reference cohort that I just mentioned earlier. So one key interest here is trying to understand the molecular characteristics driven by the tumor activity and how that is shaping the patient outcome. But one tricky thing here is that we know the brain development process really has a huge impact on the molecular profiles. And we also know that the brain development process has a close interplay with the tumor activities in those HGG tumors in the pediatrics and AYA. So therefore, we really want to dissect this complicated relationship, trying to understand what are the molecular characteristics driven by the tumor and what are the molecular characteristics that can explain by the interplay between tumor and the brain development. For that purpose, we employed a functional analysis strategy we decompose the molecular data into two parts. One part is trying to characterize the developmental process. The other part is trying to characterize the orthogonal information in the molecular profile to the developmental process. Okay, so using one gene as an example, IDH2, the left figure is illustrating how the protein abundances could depend on the age of the patient. Despite the large heterogeneity across individuals, if we fit a population level kind of functional function we can see that the protein abundances on average has a trend of increase with age in the AVA, in the pediatrics and AVA age span, while decrease in adulthood. So we call this population trajectory curve as the developmental trajectory curve in tumor, and we term it DTT. And so that helps us to characterize how the developmental process has their influence on the molecular profile. And at the same time, we also try to measure the residual, that means the distances from the observed protein abundance in a particular tumor to this population mean curve. And we term that as the residual residual to the developmental trajectory, and in short, RDT. So we will decompose the molecular profile into this two-part, DTT and RDT. And the DTT characterizes the developmental process, and the RDT characterizes the orthogonal information in the molecular profile to the developmental process. And we will try to using these two components to understand those tumors. Okay, so let's first look at the DTT matrix, which is the developmental trajectory. So this heat map is basically showing the DTT matrix derived from the RNA data, from the global proteomics data, and from the phosphoproteomics data. And not surprisingly, for most of the genes, their molecular profile has a strong dependence on age. Either they are monotone decreasing, monotone increasing, or actually some mountain shape or valley shape. So with this interesting structure of data, if we cluster samples using this DTT matrix, we actually got 4 distinct groups. While the breakpoint of age 15 and age 40 kind of consistent with the traditional definition of the AVA group, we find another significant age cutoff at 26. We refer the age segment from 15 to 26 as adolescent, and then from 26 to 40 as young adult. So this top panel is summarizing the average survival time for the patient from a given age at a given age. This is a curve of age that's based on our reference cohort survival data. And we can see that this young adult group really has the best survival compared to all the other age group. And this is also supported by our Cox regression analysis, where if we look at the KM curve of this different age group, the young adult group is significantly has better survival than adolescent, while the adolescent has significant better survival than adult or pediatrics. So this suggesting using the trajectory, we seems to be able to further dissect clinically meaningful age segments. And then if we dive into the proteomics data, we can further see that the protein trajectory actually has 4 major types of shape. A valley shape, a group of protein has a valley shape, which is enriched of peptide chain elongation. And another group of protein has a monotone increased shape, or monotone decreasing, or a mountain shape. So one interesting question we would ask is, okay, we see those trajectory in the tumor tissue. What about the normal tissue then? Will those proteins have similar or different trajectory in the normal tissue? To answer that question, we leveraged the 2 normal brain tissue data sets, the RNA data set coming from the brain Spain study, and a proteomics data set coming from actually a research team from Mount Sinai. And using the molecular data from the normal tissue, we will be able to derive a similar developmental trajectory in the normal tissue. And then we will be able to compare how the trajectory in the tumor tissue may differ from the normal tissue, and then calculate the distance between them. Okay, so this is summarized in this figure. On the left side, we see the trajectory for male, either male patient or just a male normal brain. And we can clearly see there are a group of proteins. Its trajectory is different in tumor than normal. Like in tumor, it's monotone increase, but in the normal, it increases and then decreases. If we calculate the distance between these two trajectory, which is shown in the panel on the right, we can see that this is a group of proteins has relatively higher trajectory distances between tumor and the normal. And if we further do a pathway enrichment analysis, we identified many interesting pathways having significant different trajectory distance between tumor and the normal. For example, this light blue one is actually the oxidative phosphorylation, and there are a few more. This figure is just a more comprehensive list of pathways that showing significant tumor normal differences based on their DTT matrix. We see more signals in the protein domain compared to the RNA, and we see more signals in the male samples compared to the female samples. And this really gives us some nice insight on what could be the biological processes or pathway activities that's probably reflecting the interplay between brain development and the tumor activity. Okay, I also want to comment on the other component, which is the residual part. So if we're using the residual RDT matrix based on the proteomics data to cluster samples, as you can see here, we are able to cluster samples into 4 different groups, which is each column is a sample. So as you can see, there are 4 different colors of tumor group. And we can also color proteins into 2 groups. So here we are actually using a harmonized data that's combining the whole cohort spanning from pediatrics to AYA, together with our previous CPTech GBM study, which has more adult tumors. And we are considering tumor samples from our age span from 0 to 62, actually. And as we can see here, amazingly, this protein subtype based on the RDT is show very nice concordance to the original molecular subtype of the GBM samples from earlier studies. So for people who are familiar with the adult GBM studies, we know that starting from TCGA, there has been some molecular subtypes being detected and consistently confirmed across multiple cohorts. It was named like proneural, mesenchymal, and classical subtypes. And here we are kind of surprisingly to see that after we remove the developmental process in the molecular profiles, those pediatrics and AYA tumor start to show the same molecular subtype as the adult GBM tumor. So this is quite surprising. Of course, I still want to say that there are some distinction, like for example, this IDH tumors with IDH mutation actually didn't cluster by its own, as people usually see in the adult domain. Instead, we have 2 kind of mesenchymal subtypes. One is enriched of IDH mutated tumors, and the other is basically IDH wild-type tumors. In terms of enrichment, we also see some commonalities between what we see here versus our previous understanding and knowledge of the GBM molecular subtype. Due to the time limitation, I will not go into the detail. But again, I want just to highlight that based on this observation, it certainly appears that the development has been obscuring some commonalities between tumors from different age groups that can be removing this, that by removing this effect, we can see similarities that were not apparent before. So this is exciting and may motivate alternative treatment strategies. For example, if treatment known to be effective in one age class may be successfully applied to the similar tumors in other age class. And also, as I mentioned earlier, for the AYA age group, we know the patient there has very good survival. If we know the tumors there actually intrinsically is similar as the adult tumor and the pediatric tumor, then it will be very interesting to ask what kind of brain development interplay with the tumor actually causing or resulting such a beneficial survival outcome. And the knowledge or insight on those questions probably can help us to come up with more creative treatment strategy. And we are also further extending the study by looking into some of the more recent adult GBM subtyping work, like the more recent proliferative, progenitor, and neural subtypes, which reflects a little bit better on the clinical outcome of the GBM tumor. So this is some exciting ongoing work. Okay, in the end, I want to mention that we really look to integrate many other modality of data into the picture, that the kinase analysis is ongoing, methylation analysis is ongoing, and we are also want to use network analysis to integrate different omics data. And so we are hoping that in the near future, we will be able to really understand and review many of the important insights of those tumors, this HGG in the pediatrics, young adult, and adult age span. So in the end, I want to emphasize again, this is really a teamwork. A lot of people contributed to it, and it's really a benefit to being able to have multiple consortium and to putting together expertise and resources to make those scientific work possible. And also, I want to thank the patient and patient family who contributes the samples to make this work possible. Okay, thank you. I will stop.
Thank you, Dr. Wang. That's an extraordinary talk, and I couldn't help but notice in the last 2 years, 12 cell, cancer cell, or cell reports papers. Absolutely spectacular work, and we're delighted that you've been able to join us live. So hopefully wait for the microphone to come to ask questions. If you can't hear properly, Dr. Wang, just tell us that and we'll try and paraphrase and relate the question to you. So to the audience, I've got David up there. Sorry, you're a little in the dark, David. So.
Yeah, thank you. Fantastic talk. I have two questions. One is just the survival that you showed for the older patients seemed remarkable. I don't think any of us think any of our GBM patients survive that long. So was there validation was that these were actually GBM histologically and not some other sort of misdiagnosed tumour? And the second question is, I think you compared the proteomics to your RNA-seq sort of clustering, but did you compare it with the DNA methylation profiling?
Hello, can you guys hear me?
Yes, we can.
Oh, wonderful. Yeah, thank you for the good question. Yeah, so for the first question, I assume you are asking for the second part of the talk, right? The
That's
bigger
right. Yeah,
reference data set that we pulling together, which covers a big age span. We are focusing on high-grade glioma, so including grade 3 and 4 gliomas. So, and for the adults, indeed, most of them actually is GBM. But in the adult group, I believe on that slide, I'm showing age span from 40 and above, right, as the adult group. Even within that group, actually, we can see the survival differs. as age grows. It's like for the elder patient, the survival is much worse. Like for the age span 40 and 60, actually the survival is better. Probably that is one of the reasons that the overall shape may look a little bit better than some of the other more adult-focused cohorts.
Thanks.
And yeah.
Other questions?
Yeah, just a question about the clustering that you showed with the proteomic profiling. How did that compare with DNA method, or did you look at how it compared with DNA methylation profiling?
That's a wonderful question. In the first project, we actually don't have the methylation data yet, so we didn't do that. But actually, that is ongoing effort. The methylation data is currently being generated for the cohorts in our original pediatric cell paper. So hopefully, we will have some follow-up study to be able to look into this. And for the HOPE study, yes, we are getting the methylation data, and we just finished the pre-processing, so we will look into that.
Thank you.
Thank you.
Well, while I'm waiting for others to come up with questions, can I ask, I was scouring a lot of your slides to look for evidence from the phosphoproteome of receptor tyrosine kinase activation, because I think we often see in RNA or in copy number profiles extra copies of some genes like PDGFRA, but I think really receptor tyrosine kinase activation indicated by the phosphoproteome seems quite rare. Is that true, or do you find evidence for RTK activation? Nice targets, nice drugs, but if it's not on in the tumour, not much help.
Yeah, that's a wonderful question. I think it's really just due to the time constraint. I didn't show much of the phospho data, but we definitely see signals in the phospho data. And I think but I cannot remember on top of my head the activity of each kinases across the 7 histology type. I think in the paper we did report that. And the kinase activity is very different across different histology type, first of all. And also even in the Cranio, we said we have the 2 novel subtypes. And definitely a lot of kinases like in the MAPK pathway and mTOR pathway differed between them. I can't remember whether so some of the other the players that showed differences between those 2 subtypes. We can but we should have provided all the kinase activity profiles in the supplementary table of the paper.
Yeah.
So hopefully that can allow the community to look into that in more detail.
Yes, that's a good answer. I should go and look myself, shouldn't I, in the data, which is fair enough. We've got another question from Neve.
Thank you for a fantastic talk. Can I ask about your craniopharyngioma data, some of that really nice work that's now come into the clinical trial through PNOC? So the BRAF V600E, I assume, is more common in the papillary craniopharyngioma. Are you seeing that MAP kinase alteration across the more common pediatric variant, so the adenomatous craniopharyngioma as well? And is that how that work has been translated across to the clinical platform?
That's a wonderful question, and I'm sorry I don't have a good answer for that yet. The clinical trial work I mentioned just started, and we just started to collect samples and started to settle on the experimental, like a correlated experiments that we can examine certain protein and pathway activities in them. So hopefully, like in half a year, we will have better answer for that question.
Fantastic, thank you.
Yeah.
All right, one last question, Dr. Wang, before, and it's Raylene.
Hi, thanks so much for the beautiful talk. It's quite striking, the clustering of the phosphoproteome and the proteome over the age span, over life course. I wonder how much that has to do with just the brain microenvironment, and if you did that for normal brain, would the clustering be similar?
That is a very good question. So we did compare from the trajectory angle, the tumor brain and the normal brain. And we did see the trajectory actually looks for maybe, I would say 30% to 40% of the proteins very different between tumor and the normal. Whether that is due to the microenvironment, like the cell type composition, that is still a question to be answered. We have performed immune landscape investigation for the tumor sample, like in this HOPE study already. We definitely see different immune subtype, which for some of them overlap with the molecular subtype that RDT derived subtype I just showed. But we haven't compared that with the normal data set yet. So again, I think that is a good question. It's definitely something we should look into more.